Hello everyone, welcome to your biology class. In this video, we will discuss the remaining topics of the transcription. We are already done with which strand of DNA undergoes transcription, the enzyme that participates in this process, and also about the transcription unit and various components of it. Okay, so in this video, we will study the steps of transcription process. So whenever it has to happen, all the necessities that are being explained in the previous video, they will be there and it will be carried out. The first step of transcription is initiation. The second step of it is elongation. And of course, the third and the last step, it is termination. So we will study one by one each of them. Initiation process is a one. Suppose let's draw the strands here first, okay? So these are the two strands of DNA and they must be intertwined with each other. Means they will be compactly coiled, okay? So one strand of course has to be 3 to 5 prime. And another has to be 5 to 3 prime because the two strands are anti-parallel to each other. So whenever initiation process has to happen, what will happen? The sigma factor of the DNA dependent RNA polymerase, I would really appreciate if you will come to this video after watching the part 1. Okay, sigma factor was described there. So sigma factor is a part of RNA polymerase, DNA dependent RNA polymerase that will identify the promoter sequence. And I already told you that promoter sequence is a one that has a special conserved sequences like the Tata box is found in the promoter region. Promoter region contains the Tata box. Tata box is a region where the bases are rich in T and A. So generally it has the sequence of Tata AT, means TA, TA, AT bases will be richer there. So sigma factor will come, it will bind to the place where it will find the Tata box. Now once it will bind, it will invite the core enzyme to please come up. Now when sigma factor has already bound, core enzyme has came and it will bind to the sigma factor. What will happen? Functional RNA polymerase will be formed. Okay. Functional RNA polymerase is required because this is the enzyme that will do the process. So once RNA polymerase has bound, suppose this is RNA polymerase that has bound. Now what will happen as a result of this? The DNA will get unwound. Once it will get unwound, then only transcription can happen, right? So this is the DNA and in between what will happen? A transcription bubble will be formed. Means a certain part of DNA, it will get open. And this structure which is there in the center which has opened, this is known as transcription bubble. Okay. So in initiation step, DNA dependent RNA polymerase binds to the promoter sequence. Fine. Now after this, what happens? The next steps come and this is known as elongation. Elongation is just giving an idea that the real transcription will be done in the second step. So RNA polymerase which is here, this RNA polymerase shown to you by the yellow color, it will move around towards the 3' end, okay, towards this end, okay, towards this side it is going to move. And which strand will be used this time? Template strand, okay. So in the elongation process, RNA polymerase will move towards the downstream region. And it will pick up the nucleotides from the nucleus only because this process is happening in the nucleus. So the site of transcription is nucleus only. Right? So RNA polymerase will move towards the downstream region. It will take the nucleotides, and what will happen as it is going to move further? Let me make another diagram. So this is our supposed transcription bubble, and RNA polymerase has reached here. Okay, this is the region where RNA polymerase has reached, and it has the polarity to synthesize messenger RNA in 5 to 3 prime direction. This is also very important, that RNA polymerase, it moves, it reads the bases of the template strand, complementary bases are attached, and the sequence, the single stranded RNA, it comes out. So here you can see that this is the single stranded RNA, it is coming out. Now we will see further, this is not the messenger RNA. The first form product of the transcription, it is known as heterogeneous RNA. We will be talking about it just a little later, okay? So in elongation, RNA polymerase moves and it reads the base sequence of the template strand. As per the complementary base pairing rule, the bases are attached and the single stranded RNA product, it comes out. Now once all the bases are read from the transcription bubble, that means the transcription process is almost done. So the last step that is going to happen now is the termination. Termination means the end to transcription, because whatever was required, it has already been done now. Now, transcription in prokaryotes is different and in eukaryotes it is different. In prokaryotes, it happens by the utilization of Rho factor. This is important to learn. Rho factor participates in the termination process of the transcription, whereas in eukaryotes, poly A site is seen at the terminating site. Okay, so we have seen that transcription unit has 3 parts: promoter, structural gene, and the terminating end. The terminating end would be the one that will help in the termination process. So in prokaryotes, there will be a protein which is known as Rho factor that will get associated and bring up the transcription end, right? Whereas in the eukaryotes, poly A means many adenine bases will be found in the terminating site, and this will bring an halt to the transcription process. Remember, sigma factor was a part of DNA dependent RNA polymerase that was helping in the initiation process. And here Rho factor is the one that is helping out in the transcription process. So we are done with all the 3 steps of the transcription. Now our product is ready and we will be studying about this product. Okay, so the freshly from product by transcription is known as heterogeneous RNA. This is abbreviated as HNRNA. H has to be small, N has to be small, and RNA will be in caps lock on. Remember, whenever we are writing double-stranded DNA, we need to write D small, S small, and D and A capital. Whenever we are writing RNA, it is single-stranded structure, so we will write SS. S for single, S for stranded, and RNA in caps lock on. Now this heterogeneous RNA, it is still at its site of synthesis, and its site of synthesis is nucleus only. Now you know that nucleus contains nuclear pores. From these nuclear pores, this RNA will come out. But heterogeneous RNA is not in a condition to come out, because if it will come like this, it will be digested by the cytoplasmic enzymes. You know that all this is happening in the nucleus. The contents, they have to come out in the cytoplasm because RNA has to do its further job, and that is the synthesis of the proteins. But first, heterogeneous RNA will be transformed into messenger RNA. So during this process, heterogeneous RNA, when it gets converted into messenger RNA, three things happen, okay? First is splicing. Second one is capping. And third step is tailing. So once these three steps are done, that means the heterogeneous RNA is now converted into mRNA. It is ready to go into cytoplasm because it will not be attacked by the cytoplasmic enzymes, and it can further help in the translation process. So let's see what is splicing. The heterogeneous RNA that is being formed, right? It has coding and non-coding sequences both, okay? So here you can see that I am making the strand by the use of two different colors, okay? This is our 5' end, this is our 3' end, and still this is our heterogeneous RNA because it has not undergone processing. Remember, the sequences in red, they are the ones which are important to us, and these are known as exons. These are the ones which are important, thus these will be counted as the coding sequences. Whereas the black sequences are the introns. They are intervening the exons, right? So we can call them as intervening sequences as well, and they are not important to us because they will not participate in the translation process. So what is the use of them? It's better to get them removed, and that is what our splicing process is. So splicing refers to the removal of these introns and ligating or joining all the exons together. Okay, so if you remember, I said in the previous video that snRNA is a type of RNA which is produced by RNA polymerase III in the case of eukaryotes, right? So this snRNA will help out in the splicing process. It will bind to introns in this manner, right? So this structure, this yellow structure is made by the snRNA. And what will happen? It will take them out. So in the second step, what is going to happen? These sequences will be taken out by snRNA. You can see that introns are now being removed in step number 2. Now in the third step, you can see that ligation has to happen. Ligation means joining has to happen because all the exons, they should be present in a stretch. So this is our exon 1, this is exon 2, this is our exon 3, and these exons will be ligated. So this is our E1, exon 1, exon 2, exon 3, and overall you can check now that all the coding sequences, they are present in a stretch. So this is what splicing is. Splicing is the removal of all the intervening sequences or introns and the ligation of all the exons or coding sequences together, right? After this, the second step happens, and that is the capping step, okay? Capping means, of course, this structure which is formed here, it will be wearing a cap. It will be wearing a cap because wearing cap is important, otherwise the enzymes of cytoplasm, they are going to break it. So it will wear a cap, and the chemical called 7-methyl-guanosine, right? 7-methyl-guanosine, it will be added to the 5' end. Means here at 5' end, this is going to attach, and this will act as a cap so that enzyme should not recognize it, and it will be protected. It will further do its part. Now capping is also done. We are left with tailing, which is the last step of processing of hnRNA. Tailing means tail will be added. So this is the end, 3' end is the end. At this end, now certain bases will be added. Okay, so at the time of tailing, polyadenylation is done. Polyadenylation means the addition of the A base, adenine base will be done, right? So I am writing here only this will be removed, and what will happen in place of it, A sequences will be added. So this will become the 3' end. So ultimately our product will be a cap in the beginning, which is 5' end, all the exons together, poly A sequence at the 3' end. When all these 3 steps are done, one after the other, means splicing, capping, and then tailing happens, then heterogeneous RNA is known as messenger RNA. This messenger RNA is the one that comes out of the nuclear pore, remains resistant with enzyme attack, and helps in the translation process. Before I end up, I just want to explain one more term, and that is monocistronic and polycistronic. Many times it has been asked in the board examination difference between the monocistronic and polycistronic genes. So remember, eukaryotes, they are monocistronic in nature because they contain monocistronic genes, whereas the prokaryotes, they contain polycistronic genes. Now you know that this messenger RNA is formed, right? So if you have one messenger RNA and if it is synthesizing just a protein, means one protein, then it is going to be monocistronic. Okay, this is the case in the case of eukaryotes. But when one messenger RNA is there and it is synthesizing many proteins, then the case is known as polycistronic. This we will see in lac operon in the case of bacteria. If you are already knowing lac operon, then you must be knowing that there were 3 structural genes, Z, Y and A, and all of them were synthesizing the proteins from one messenger RNA only. So that was polycistronic in nature. We were studying in bacteria. Bacteria is an example of prokaryote, right? So eukaryotes contain monocistronic genes, and bacteria or the prokaryotes, they contain polycistronic genes. I hope this has been understood. In case of any query, you may leave a question in the comment box. I will revert to you as soon as possible. And if you haven't subscribed to my channel, please do so and share with maximal friend of yours so that they can also be benefited. Thank you and stay tuned for more videos.